Okay, let's have a quick look at Punnett squares. Now, before we do that, we need to just clarify what we mean by genotype and phenotype. Now, hopefully, if you're looking at Punnett squares, you already know what genotype and phenotype are. So, your genotype is the combination of alleles, right? It's basically the letters. So, we have these alleles, a version of a gene, and the combination that you have is your genotype. So, just as a quick example, it might be big T, might be big T, it might be little t. That is one genotype, that is another genotype, that is another genotype. Okay? Now the phenotype is what you actually look like. It's the actual trait that gets expressed. So it's the trait expressed, let's put it that way. Genotype says, this is what you should look like. Phenotype says, well, this is what you actually do look like. So if we're using the same example from the genotype, I used T's, I was just thinking tall or short plants, right? Like from Mendel's peas experiments. So the phenotype would be either a tall plant or a dwarf plant, a shorty. Okay, so the genotype, the big T that says, hey, I'm pointing towards the tall phenotype, okay? And the little t says, hey, I'm pointing towards the dwarf phenotype, to be a short plant. Now, remember, we don't say that alleles are dominant or recessive. We can't say that the big T is dominant to the little t and the little t is recessive. No, we can't. What we can say is that the tall phenotype is dominant to the dwarf phenotype, to the short phenotype. Okay, so that's just a quick refresher on genotype and phenotype. All right, genotype is what the genes say you should look like. So you've got two versions of the gene called the allele. And, you know, in this first case, we've got two big T's. So both alleles say, hey, you should be tall. In the third example here, we've got both alleles saying, hey, you should be short. Now, in the middle example, we've got a big T and a little t. So we've got one allele saying, hey, you should be tall, and the other allele saying, hey, no, actually, you should be short. What are you going to be? You're going to be tall, because tall is dominant. If you're, I could be tall or I could be short, well, tall is dominant, so you'll be tall. Remember, with Mendel and his peas, with Mendelian genetics, it was always a case of complete dominance, one version over the other version, okay? Which, of course, is not always the case in real life, but that's okay. It makes it quite helpful for our Punnett squares, which we're now moving onto. Okay, so a Punnett square is a helpful way to predict the possibility of the offspring having certain characteristics. It does not guarantee that the offspring is going to have certain characteristics, but it is a good way to make a prediction. So let's say we cross, how about we stick with the same example that we just mentioned, right? Tall or dwarf plant. So we're looking at Mendel's peas. Let's say one plant was homozygous. Okay, there's maybe another term we should just recap. This is homozygous, so is this. Whereas this one is heterozygous. Okay, so let's just break those apart. Homozygous, both the same, right? So homo meaning same, these are homozygous, and hetero meaning different. Hetero, different, is a hetero, well, this is heterozygous. So you've got two homozygous versions and one heterozygous version. So homozygous, homo, same, yeah, because both letters are the same. They're both, in this case, capital, or in this case, they're both lowercase. Hetero meaning different, because our alleles are different. We've got a capital T and a lowercase t. So they're different, right? Different terminology we can use as well. Homozygous, we can say is pure, or pure breeding, or purebred, and heterozygous, we call them hybrids. Okay, so different terminology meaning the same thing. Okay, cool. Now that we've got our letters taken care of, let's actually do a Punnett square. So where were we? We were starting with crossing a homozygous. Okay, so both letters are the same. Homozygous dominant, so both of these alleles point towards the dominant trait. Let's cross it with a homozygous recessive. Okay, so again, homozygous, both letters are the same, and both of these alleles are pointing towards the recessive trait. What are the chances of getting a short plant? as an offspring? Well, here's what we do. This is called a Punnett square. We draw up a grid. Okay, now across the top, we put one parent. Down the side, down the left-hand side, we put the other parent. So let's do the first parent. That parent could have given a capital T, or it could have given a capital D, right, if it gave you the other allele. Remember, you get one allele from each parent, right? So the sex cells are haploid, but then, of course, the organism is diploid, right? So we have to end up with two versions of the gene, one from mum, one from dad. Okay, so dad could either give us this one or this one. Now, in this case, they're both the same because dad was homozygous, right? And from the other parent, we get a little t or we get a little t. So we're getting one allele from each parent. What is bub going to look like? Well, if we're talking this square here, then bub is getting a big T from one parent and a little t from the other parent. And it really doesn't matter whether you write the male across the top and female down the side or vice versa. It doesn't matter, right? One parent and other parent is good enough. Now, what about the, maybe we have a baby with this genotype. What's that genotype going to be? Well, that's going to have a big T and a little t. And down here, we're going to get a big and a little, and here we're going to get a big and a little. So all possible outcomes for the genotype, all offspring are going to have, so we've got all, or I guess you could write 100%, all genotypes are going to be big T, little t. Okay? Now, what does that mean in terms of phenotype? They will also be all, right? Or you could do 100%. They're all going to be tall plants. Okay, so the genotypes, they're all big T, little t. In terms of the phenotype, what do they actually look like? Well, all of these plants are going to be tall because all of them have a big T. Now, the big T is pointing towards the dominant trait, and we said the dominant trait in this case is tall. So all of the offspring will be tall, 100% of them. Okay, let's look at another example. So, in another example, let's talk about cabbage butterflies. In cabbage butterflies, white wings are actually dominant to yellow wings, right? So let's go white. Let's just write down the information. White is dominant, and what was it? Yellow. Yellow is a recessive trait. So white is the dominant trait, white wings. Yellow is the recessive trait. What letter are we going to use? Well, we're going to use a W. So remember, we tend to use a letter that corresponds to the dominant trait. Okay, well, I'm looking at white being dominant, so I'm going to use a W now. So I'm going to use a capital W for the dominant trait and a lowercase w for the recessive trait. Right. Now, let's do a cross. Let's say a heterozygous butterfly is crossed with a homozygous recessive. All right, let's just do that one. So we're crossing a heterozygous, so hetero, different alleles, right, one of each. We're crossing that with a homozygous recessive. So homozygous, both letters will be the same, but recessive, so they'll both point to the recessive trait. So we're looking at the little W's. And then we draw a Punnett square. So here we go, Punnett square time. Once we've got our Punnett square, we throw down what each parent could possibly give. Well, the first parent could give a big W or a little w. The second parent could only give a little w in both instances. And then we can say, okay, what are the offspring going to look like? Well, the first offspring will have a big and a small. So we're just matching them. With this Punnett square, we match from across the top with down the side. So we start at the W and we start at this W and we just come down and we come across until we match them together to get a big and a small. And then we start at this one and we start at this one on the side and we match them together and we get two smalls. And likewise, we have two little W's, a big W and a little W. Great, so there's our Punnett square filled out. Now, what does this actually mean? Well, we can talk about the genotypic ratio and the phenotypic ratio, right? So the genotypic ratio, you can see we have 2 big, small to 2, I need to do it big, to 2 small. All right, now, of course, it's a ratio. We would simplify that, would say is 1 to 1. Okay, so 1 big W, little w to 1 little w, right? There's our genotypic ratio, or of course we could do it percentages. We could say 50% of this genotype and 50% get this genotype. So remember, genotype is the combination of alleles. It's basically what letters do you have. Now the phenotype, the phenotypic ratio, well, that's also going to be 50%, right? It's 50-50 or 2-2 or 1-1. But the phenotype is what does the organism actually look like. It's what trait actually gets expressed. Well, in these two on the left, we have a capital W, so those butterflies are going to have white wings. So we're going to have 50%, right? So because 2 out of our 4 possible offspring, 50% white wings, and we will get 50%, the other 50% will have the yellow wings, the recessive trait. Okay, so there we go. We use a Punnett square to work out the genotypic and phenotypic ratios, or what are the possible genotypes, what are the possible phenotypes. Great, let's do another example. Okay, let's look at guinea pigs. Now, guinea pigs, short hair is dominant over long hair. So we're going to use S for short, because short being our dominant trait, that's going to give us the letter to use, right? So we're not always using A's or B's or C's. We just make the letters kind of match what trait we're talking about. So short is dominant. We're going to use S. So let's cross a homozygous dominant guinea pig. So we've got dominant, there we go, and homozygous. So homozygous, the letters are the same. Dominant, we've got the alleles going towards the dominant trait. Let's cross this with what should we cross it with? A long-haired guinea pig, a homozygous for long hair. Okay, so short is dominant over long. We're crossing two homozygous parents, one homozygous for the dominant trait, one homozygous for the recessive trait. We draw up our Punnett square. There we go. We change colour because we can use multiple colours now. And one parent will give us a big and a big. The other parent will give us a small and a small. What are our possible combinations of those? Hey, go away. There we go. Combinations are a big and a small and a big and a small and a big and a small and a big and a small. So we can see that this is very much like our first example, right? Remember the first example, we actually did have homozygous dominant crossed with homozygous recessive, and they were all this genotype and they were all this phenotype. Well, I just did this off the top of my head and we have arrived at the same result, because in terms of our genotypic ratio, 100% of the genotypes are big S, little s. And in terms of the phenotypic ratio, 100% of the phenotypes of the trait that actually got expressed, they've all got a big S, so they're all going to be, what did we say was dominant? Short. They're all going to be short. Short-haired guinea pigs for all of the offspring, right? Now, here's another word, a carrier, because let's say this guinea pig here, this guinea pig was born, and it's a short-haired guinea pig. Yep, cool. It could have a baby with long hair, because it has the allele pointing to the recessive trait, to the long hair. That trait just doesn't get expressed because there's also an allele pointing to the dominant trait, right? But the allele is there. So when this guinea pig had a baby, maybe it gives this allele to the bub, or maybe it gives this allele to the bub. And if it gives the little s, and the other parent gives the little s, then the baby will be a long-haired guinea pig, right? So this guinea pig has short hair, but we use the word carrier. It's a carrier of the gene for long hair. That gene is not expressed, but it's sort of hidden there. It's masked there. But it could come out in this offspring's offspring, right, in the next generation. Let's do one more example of this type of Punnett square. This type of Punnett square is called a monohybrid cross. We're only looking at one trait at a time. Okay, we can also use Punnett squares for a dihybrid cross, where you look at 2 traits at a time, or a trihybrid cross, where you look at 3 traits, and they just get more and more complex, right? So this is the nice fun version, although I quite like the dihybrid crosses as well. I think they're fun. But anyway, let's look at tongue curling. Some people can curl their tongue. I, myself, and other people can't. So let's cross a tongue curler, heterozygous, with another tongue curler who is also heterozygous. Okay, so in people, tongue curling is a dominant trait. Okay, so we're talking humans here. Being able to curl your tongue sideways, up around the sides, that's a dominant genetic trait. So both of these parents are heterozygous, or we could say they're both hybrids, all right? And they are both tongue rollers because they have an allele that says, hey, you should have the dominant trait. And so sure enough, they do have the dominant trait. Cool, so let's do that cross, and we draw up our little Punnett square here. Chum chum, fill in that hole. Right, so one parent could give a big T or a little t. The other parent could give a big T or a little t. What are the possible options that we have for our 4 offspring? Well, the first one could be big T, big T. The next one could be big and a small, and then we have a big and a small, and then we actually have 2 small. So we have 3 different genotypes here. Only 2 phenotypes, but we've got 3 different genotypes, right? So let's look at our possible options for the offspring. In terms of the genotypic ratio, we have 25%, or we could say 1 to 2 to 1, or 25%, 50% to 25%. But 25% of offspring are going to be big T, big T. Right? And then we've got 50% of the offspring are going to have 1 big T and 1 little t, and the other remaining 25% will be both little t. Okay, so 3 options for a genotype, because we've got this version, or we've got this version, or we've got this version, right? 2 of those, so 50% of that version. Now, phenotype. We actually only have 2 options now for the phenotype, because remember, if you have 1 allele pointing to the dominant trait, you're going to have that dominant trait. Well, here we've got at least 1, definitely. We've actually got 2, but we've got at least 1. Here we have at least 1, and here we have at least 1. So these 3 babies are all going to be tongue rollers, because they all have at least 1 allele pointing towards the dominant trait. So our phenotypic ratio is going to be 75%. We're looking at now 3 to 1. Okay, 75% are going to be tongue rollers. They can roll their tongue. And the 1 bub left over, the 25% remaining, is not. Right, is not going to be a tongue roller. Now, keep in mind, this is all statistical probability, right? Let's say these 2 parents actually have 4 children. We're not necessarily going to get 3 of those children are tongue rollers and 1 isn't. Maybe none of them can roll their tongue. Maybe 50-50. Maybe only 1 can roll their tongue and 3 can't. Okay, so the Punnett square doesn't tell us what we're actually going to get, but it tells us the probability that we could possibly have. Okay, so Punnett squares don't tell you exactly, they tell you your chances of getting certain genotypes and phenotypes. So those were monohybrid crosses. Let's take a look at a dihybrid cross, where we look at 2 traits. Okay, so first we're going to need to come up with some traits. What are some traits? Oh my goodness me, I don't know. Let's look at widow's peak versus non-widow's peak. If you're not sure what a widow's peak is, here we go, let's draw some people. Let's give them some hair. Whew, awesome drawing skills here. Good job. So there's the hair. Now, the person on the left, their hairline goes straight across. They do not have a widow's peak, right? No widow's peak with their hairline. We're talking the front of their hairline up around the forehead. The other person, the hairline sort of comes to a bit of a pointy part in the middle. That is a widow's peak. All right. So there we go. Widow's peak, by the way, in humans is dominant. All right. So if one of your parents have a widow's peak and they are homozygous, then you are guaranteed to have a widow's peak. If one of your parents has a widow's peak, doesn't necessarily mean that all the babies will, because they might be heterozygous, right? They might be carriers for the gene to not have a widow's peak, right? So widow's peak is the dominant trait, all right? So, all right, let's look at widow's peak. What else shall we look at? Off the top of my head, can skate or can't skate. All right, so the second trait we're going to look at is, I nearly do the skateboard. Let's give this person some legs. There we go. So that person is a skater. This person can only walk because they don't have a skateboard. So now this is of course not a genetic trait. This is, hey, do you practice and do you learn it? But off the top of my head, let's just look at this. Right? Let's pretend that this was a genetic factor of whether or not you were a skateboarder. Now, which one should we make dominant? How about not skateboarding, because not skating is easier than skating. So let's say, that didn't quite work out. Let's say that not skating is the dominant trait. Okay, cool. So now we'll do a cross. So what shall we cross? Let's cross a widow's peak, heterozygous. There we go. Oh. not H for heterozygous, but a small w, right? A big and a small. So we'll cross that. Now, what about the other trait? Are they a skater or not? Flip a coin, no, they're not. Okay, so no, they're not is the dominant. So we'll say not skater, and let's make them heterozygous for that. There we go. Let's cross this with a person who doesn't have widow's peak, so little w, and can skate. Now, shall we make them heterozygous or homozygous? Let's make them heterozygous. So we are heterozygous for... Wait, hang on. Oh, no, they have to be homozygous. There we go. Because not being able to skate is a dominant trait. So that means if you can skate, you actually have to be homozygous for that trait, right, for the recessive trait. So there we go. Okay, so what we're talking about is the first person, one parent, has a widow's peak because they've got a big W. Cannot skate because they have at least one of the alleles that points to the not being able to skate. The other parent does not have a widow's peak because they are homozygous for the recessive trait. And they can skate because they are, again, homozygous for the recessive trait. Right? Dominant being can skate, so the recessive trait can't skate. Let's see what the children of these people come out like. So our Punnett square, in this case, is just going to get a little bit bigger, since we're looking at two traits at a time, right? Oh, hang on, I need even bigger. There we go. So we're looking at two traits at a time, so our 2 by 2 suddenly jumps up to be a 4 by 4, but that's fine. One parent across the top, other parent down the side, right? That part hasn't changed. So let's put the first parent along the top. So this parent could give a big and a big. Okay, so we're looking at two traits at a time now, so we have to write two letters. So this parent could give a big and a big. Or that parent could have given a big and a small. So they could have given a big W but a small n. Okay, all right, now that parent, maybe we don't start with the big W, maybe they gave the little w and the big N. Or maybe they gave the little w and the small n, right? Little w and small n. Okay, so above each column we actually have two letters, right? Because we're looking at two traits. And then we just do the same for the other parent. So the other parent, well, it's all recessive. So they could either give a small and a small, or a small and a small, or a small and a small, or a small and a small, right? Because we're recessive, both the alleles are pointing to the recessive trait for the widow's peak, and both are pointing to the recessive trait for the skating ability. Okay, now hopefully you can see that there's actually going to be a bit of a pattern here, because if we now match up the top parent with the down the side parent, right, the across the top with the down the side, we're going to get a big W and a little w and a big N and a little n. However, immediately below that, it's not going to change, is it? Because down the side, we've got all the same possibilities. So we don't have to really think too much to try and work out each individual cell, spot the pattern, and then we can just copy it in. All right, and this is the same, and the last one is the same. So if you can spot the pattern, that'll just make things quicker, right? If not, well, you just work it out cell by cell. It's doable. It just takes a little bit more time. So now the next one, what have we got? We've got a big W and a small w and two little n's. So big W, small w, two little n's. And of course, working down, as we just said, well, it's always little w, little n, so it's not going to change. So we have big and then small, 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 and then a big, and then a small, 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 a big, a small, small, small. Cool. And we move on to the next one. We've got two small w's, one big N and one little n. So small dub. We have a big N and a small n. Okay. Now I'll save my time and just, there we go. That carries all the way down. And then we have just everything small. Small, 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 and that repeats all the way down. Great. So I took a little bit of a shortcut there, just drawing arrows saying, hey, look, it's same. But remember, the Punnett Square is just my way to work out, right? So let's look at our genotype and phenotype ratios. Well, I'm a bit out of space, so we'll jump down here. So our genotypic ratios, we have 4 options, right? We had this option, this one, or this one, okay? So our genotypic ratios are going to be 1 to 1 to 1 to 1. Right, 4 to 4 to 4 to 4, but of course we simplify. Yeah? We could talk percentages, 25% to 25%, etc. But so we have genotypic ratio. We have 1 to 1 to 1 to 1. Now, what are we actually talking about? That ratio is actually talking about big, small, big, small. So we've got that ratio is of big, small, to, what have we got? Big and then all small. Big and then all small. And then we had 2 small, a big and a small. So we had, what did I say? 2 small, a big and a small. And our last option we have is all small. So small dub, small n, small n. So that's our genotypic ratio. Now, in terms of our phenotypic ratio, what are the actual traits of these people? Well, the ratio is 1 to 1 to 1 to 1 again, so 25%, 25%, 25%, 25%. But what are we talking about? What are those 1, 1, 1, 1s? Well, the first person, this person has widow's peak and is not a skater, right? Because they have the allele that points to the dominant trait for both of those options, for the widow's peak versus the skating ability. So this person cannot skate, so and has widow's peak. So I'll just say widow's peak, non-skater. Okay, so we have 4 of them, and we have 4 of them to, what's the next person? Well, the next person also has a widow's peak, but actually this person can skate because we don't have any allele leading to the dominant trait of not being a skater. So this person has widow's peak, but can skate. Yay, go them. The next person doesn't have widow's peak, cannot skate, and the next person doesn't have widow's peak, can skate. So the next person, non-widow, non-skate, and the last person, non-widow, and can skate. Cool. So there are our phenotypic ratios, our 4 phenotypes, and they got a bit squashed up towards the end. But, allow yourself enough room to write down all the phenotypes. So there we go. So that's a dihybrid cross. All right. So another example that that Mendel actually did with his peas, because Mendel didn't just do monohybrid crosses; he did dihybrid crosses. So he actually crossed round peas that were yellow in colour, homozygous for being round and homozygous for being yellow, and he crossed that with homozygous for being wrinkled and green. Remember, Mendel looked at opposite traits. He crossed the opposites, right? And so all of the offspring of this cross, so we call this the F0, or sometimes the P generation, P for parental, or F0, right, for filial, but we're starting at 0. And then in the filial 1, what are our possible options for all offspring? Well, a big little I and a big Y, little y. So all of these offspring were round, because they had the allele pointing towards the round, and they were yellow, because they had the allele pointing towards yellow, right? Round and yellow are both dominant over wrinkled and green. And then he selfed this one. Okay, so we did a dihybrid cross, and then this F1 generation, he selfed it to get the F2. So essentially, he crossed that with exactly the same, okay, to get the F2 generation. Now, the ratio he got here in terms of the phenotypic ratio, so phenotypic ratio 9 to 3 to 3 to 1. Okay, 9 plants were round and yellow. They had both dominant traits. 3 plants had one dominant trait and the other recessive, so they were round but green. And then another 3 were sort of the opposite, so they were wrinkled and yellow. And then one little guy, one little plant, well, 1 out of the 16. You know, if there were thousands of plants, we're not just saying one plant, but 1/16 of all of the plants were wrinkled and green. And so it kind of looks a bit like this. So there we go. That's Punnett squares, monohybrid and dihybrid Punnett squares. Sorted.
Don't forget to hit the like button. Yeah. And subscribe to It's Up Here. And here's another video.